bond is a chemical bond formed when a carboxylic acid reacts with an alcohol, releasing a molecule of water in the process. This reaction is called esterification, and the resulting bond links the oxygen atom of the alcohol to the carbon atom of the acid's carbonyl group, creating an ester. Ester bonds are very important in biology and industry. In living organisms, they connect fatty acids to the glycerol to form triglycerides, which serve as energy storage molecules, and they are also found in phospholipids that make up cell membranes. In nucleic acid like DNA and RNA, a special type called phosphoester bond link sugar and phosphate groups, helping stabilize genetic material outside the body. Ester bonds are used to make perfumes, flavors, synthetics, and materials such as polyester. An ester bond is formed through a reaction called esterification, where a carboxylic acid react with an alcohol. During the reaction, the group that the acid and the atom from the alcohol combine to release water. After water is removed, the oxygen atom from the alcohol bonds with the carbon atom of the acid carbonyl group, creating the new linkage, which is the COO, which is the ester bond. The product of this reaction are an ester and water. For an example, acetic acid plus ethanol is equal to ethyl acetate plus H2O. Why ester bond is important? Ester bond play a vital role in both living system and everyday life. In biology, they connect fatty acids to glycerol to form fats, which serve as the body's main energy storage, and they are also found in phospholipids that build cell membranes. In DNA and RNA, phosphoester bonds hold the sugar and phosphate backbone together, making genetic information stable. Beyond biology, ester bonds are widely used in industry and medicine, forming the basis of perfumes, flavors, plastics like polyester, and even certain drugs that rely on ester linkage for activation. Overall, ester bonds are essential because they support life process and have many practical applications.
And now let's talk about urea cycle.
The urea cycle, also known as the orthonin cycle, is a vital process that takes place in the liver to remove excessive nitrogen from the body. When proteins and amino acids are broken down, they release ammonia, which is highly toxic if it builds up in the blood. To prevent this, the urea cycle converts ammonia and carbon dioxide into a safer compound called urea, which is then transported to the kidneys and excreted through urine. This cycle not only protects the body from nitrogen toxicity, but also help maintain a healthy balance of nitrogen, which is important for growth, repair, and energy metabolism. In short, the urea cycle serves as the body's natural detoxification system for handling waste nitrogen, because it plays a vital role in removing harmful nitrogen waste from the body. When proteins and amino acids are broken down, they release ammonia, which is highly toxic if it builds up in the blood. To prevent this, the liver uses the urea cycle to convert an ammonia into urea, a safer compound that can be transported in the blood to the kidneys and excreted through urine. This process not only protects the body from ammonia, but poisoning, but also help maintain nitrogen balance, which is essential for proper metabolism and overall health. Without the urea cycle, the body will not be able to handle the byproducts of protein breakdown safely. The ester bond and the urea cycle are both essential for life. Ester bond play a key role in building and storing important biological molecules like fats, phospholipids, and nucleic acid, while the urea cycle ensures the safe removal of toxic nitrogen by converting it into urea for excretion. Together, they highlight how the body uses chemical process to store energy, build structures, and maintain balance for survival.